Up to now, we've been focusing mostly on MHC, on the interaction of T cell receptors with MHC or B cell receptor with an antigen or something from an APC. But that's not the only players when it comes to our immune cells. Got to remember, our immune cells are also producing cytokines and chemokines. So you're going to have receptors just for them. Because remember, it's not a generic one receptor can bind a bunch of different ligands, and depending on which ligand it is, the cell will respond differently. No, it is one ligand, one receptor. So you're going to end up with, as you see here from this figure from the textbook, remember this textbook is edition 8, which I have uploaded to Canvas. You're going to see that there are multiple different cytokine receptors that take on multiple different shapes. They have multiple different domains and motifs depending upon what they're for. Great example of this is the JAK-STAT pathway. JAK-STAT binds to certain cytokines. Remember, cytokines are going to be the things that activate new abilities. So JAK the receptor is a dimer form. The two halves don't like to play with each other until the cytokine is there. Cytokine binds the two halves, stabilizes them, and hey, look, they phosphorylate each other. STAT will then be recruited in. STAT recruits in, becomes phosphorylated, and now forms a dimer itself. The phosphorylated form forms a dimer, which just so happens also have a high affinity for specific DNA sequences. Now becomes a transcription activator. It goes, binds, and next thing you know, you have the transcription of new gene products.